Dear students, today we are going to start a new session called Bio Prime Points. This is mainly for the revision of each and every topics focusing your board examination. In today's class, we are going to discuss the essential points, the prime points of the first chapter, Sexual Reproduction in Flowering Plants. This chapter is divided into two sessions, part 1 and part 2. In this video, we are going to discuss the important points, the prime points of the part 1 of sexual reproduction in flowering plants. Okay, now we can start. The first point, the first prime point is stamens. Stamens are the functional units of androecium. The functional units of androecium are called stamens. We can draw the structure of the stamen. This is the diagram of a stamen, a diagrammatic representation of a stamen. The stamen has two parts. This is a stalk. The stalk is called filament. And at the tip of the filament, there is an enlarged bilobed structure. The functional part is present, that is called anther. Okay? So, a filament and anther are the components of a stamen. So, components are the filament. Filament is the stalk of the stamen and is long and slender. Then come the anther. Anther is the functional and enlarged tip of the stamen. Only two parts are there. Then what about the anther? It is a functional part. The anther is considered to be bilobed and dithecous. ആ രണ്ട് പോയിന്റ് വളരെ ഇമ്പോർട്ടൻ്റ് ആണ്. നോക്കൂ, in the diagram, the anther has two lobes. This is lobe number 1 and this is lobe number 2. 1 and 2. Two lobes are there. Hence it is considered to be bilobed. Bilobed means having two lobes, a right lobe and a left lobe, like that. Then each lobe has two cavities inside, two chambers inside it. That means in the cross section we can see that this is the cross section of one lobe and this will be the cross section of the other lobe. Okay, and these are the regions of anther. Okay, that means inside one lobe you can see that two chambers, two longitudinal chambers are there. And since each lobe consists of two chambers, they are called what? Dithecous. That means each lobe with two thecae. Thecae means elongated chamber. So what is the total number of chambers? One lobe has two chambers. So total number of chambers 4. 1, 2, 3 and 4. And each chamber present inside a lobe is called Microsporangium. That lobe is called Microsporangium. We can draw another section, a larger view. This is one lobe having two chambers or two microsporangia. And this is the other lobe, again with the two microsporangia. And since there are 4 chambers and each lobe has 2 chambers, it is called dithecous. So that the total outline is tetragonal, 4-sided. Okay. So anther is tetragonal in outline and contain 4 microsporangia. Then what is a microsporangium? Microsporangium is the region where microspores are produced. The production of microspore is called Microsporogenesis. The area where Microsporogenesis takes place is called Microsporangium. That means one anther consists of how many microsporangia? 4 microsporangia. So anther is a functional part of the stamen. Anther is bilobed. Each lobe has 2 thecae, so they are dithecous, and altogether there will be 4 microsporangia. So it is called a tetrasporangiate. Microsporangium is the region where microspores are produced. Clear? Now we move on to the next prime point, that is carpel or pistil. Just like stamens are the functional parts of androecium, carpels are the functional parts of gynoecium. The functional units of gynoecium are called carpel. There is one more name for the carpel. Carpels are otherwise called pistils. So this will be the diagram of a pistil. Okay. The pistil has three regions. An enlarged tip is there, it is called stigma. Then a tubular portion is there, that is called style. And the basal swollen point is there, that part is called ovary. So you see, the components are stigma, it is a landing platform for pollen grains. Pollen grains are falling on the stigmatic surface during the time of pollination. So stigma is a receptive center for pollen grains. Then there is a tubular portion, elongated tubular portion, that part is called style. Then the basal bulged part is called ovary. So three regions are there. And inside the ovary there is a chamber. The cavity present inside the ovary is called locule. Some ovaries may have a single chamber. That kind of ovary is called monolocular. Sometimes the chamber may be divided into subdivisions by partitions. So that is called multilocular. So a cavity present inside the ovary is called locule. Then ovules or megasporangia are seen inside the locule and are connected to the ovary wall by means of placenta. Suppose this is a ovule. Okay. Ovule is present inside the locule, and the ovule is connected to the ovary wall by means of a stalk, by means of a connecting structure. That structure is called a placenta. So carpel has three regions: stigma, style, and ovary. The cavity present inside the ovary is called locule. Within that locule, ovules are present, one or many, and the ovules are connected to the wall of the ovary by means of placenta. Ovule is nothing but megasporangium. Ovule is nothing but megasporangium. Megasporangium. What is megasporangium? Just like microsporangium is the area where microspores are produced, megasporangium is the area where megaspores are produced. The megaspores are produced inside the ovule. So ovule is the megasporangium. Megasporangium is the area from where megaspores are produced. Megaspores are otherwise called what? Embryo sacs. So these are the main points you have to remember regarding the structure of a carpel. Okay. Now we can move on to the next prime point. There is microsporangium. As we already stated, microsporangium is the region where microspores are produced. Microsporangium is the region of microspore formation. What is the process of microspore formation is called? Microspore formation is called Microsporogenesis. So it is called Microsporogenesis. So you can see that this is one sporangium, microsporangium. Inside the microsporangium, microspore producing tissue will be there, and the tissue is called sporogenous tissue. Sporogenous tissue. You can see here, sporogenous tissue. Sporogenous tissue means spore generating tissue. And the spore generating tissue is covered over by a layer, and that layer is called tapetum. It is called a tapetum, the innermost layer which surrounds the sporogenous tissue is called a tapetum. This is tapetum. Okay. And the tapetum is covered over by another layer. That layer is called endothecium. It is called endothecium. Endothecium is again covered over by another layer. That layer is called epidermis. So this is epidermis, this is endothecium. The innermost one is called tapetum. Now in between tapetum and endothecium, so many layers are there. So many layers are there. All these layers are collectively called middle layers. So 4 regions are there. Outermost one is called epidermis. Inner to epidermis is endothecium. Inner to endothecium is middle layers. The innermost one is called tapetum. All these are collectively called anther walls. All these are collectively called anther walls. Okay. Here, tapetum is very important. The tapetum, since it is a immediate layer over the sporogenous tissue, the tapetum is considered to be the nourishing layer. It is a nourishing layer. It is a nutritive layer that provides nutrition to the developing sporogenous tissue. All others, such as epidermis, endothecium, middle layers, they are considered to be protective and help in the dehiscence of anther. Okay, epidermis, endothecium, and middle layers are protective in function. Tapetum contains binucleated cells with dense cytoplasm, which nourishes the developing pollen grains. Okay, anther walls are epidermis, endothecium, middle layers, and tapetum, 4 layers. At the center of the young microsporangium, compactly arranged sporogenous tissue is occupied. What is sporogenous tissue? The spore generating tissue. Okay. Next prime point is megasporangium. Megasporangium is the area where megaspores are produced. We already discussed that megasporangium is another name for ovule. It is another name for ovule. Ovule is a megasporangium. Where you can see ovule is present inside the ovary. Inside the ovary there is a space called locule, and within that space the ovule is present. I will draw like this. This is the wall of the ovary. This is the ovary wall. Okay, and this is the ovule. So this is ovary wall. The cavity inside the ovary wall is called locule, and ovule is present inside it. This is called ovule. Megasporangium is a region of megaspore formation. Megasporangium is connected to placenta by a stalk called funicle. The junction between funicle and ovule is called hilum. So here there is a stalk. The stalk is called funicle. Funicle is the stalk of the ovule. The funicle attaches to the ovule at a point. That point is called hilum. The point is called hilum. And the funicle connected to the ovary wall by means of a cushion-like structure, and that structure is called a placenta. Okay, that structure is called a placenta. The junction between funicle and ovule is called hilum, and the junction between funicle and ovary wall is called placenta. Now, each ovule is covered over by 2 types of envelopes. These are the envelopes of the ovule. Outer and inner envelope. They are called integuments, collectively called integuments. The outer integument is called testa, the inner integument is called tegmen. And there is an opening at one point. That opening is called micropyle. Outer and inner integuments enclose a tissue. The tissue present inside the ovule is called the nutritive tissue. The tissue is called nucellus. The nourishing tissue present inside the ovule is called nucellus, which has abundant food reserves. Megaspore or embryo sac is produced within the nucellus. Suppose this is a megaspore. Megaspore is produced inside the nucellus. Opening at the tip of the ovule is micropyle. You see, this is a micropyle. The basal portion of the ovule, where the funicle attaches, the opposite point of the micropole, that pole is called chalaza. This is the chalaza. So suppose this is the ovule. One pole is called micropylar pole. The opposite pole is called chalazal pole. The base is called chalaza. The opened tip is called micropyle. So these are the regions of Megasporangium. Megasporangium is the area for megaspore formation or megasporogenesis. The Megasporangium is otherwise called ovule. Okay. Now the next prime point, that is the Microsporogenesis. Microsporogenesis means the process of formation of microspore. The process of formation of microspore within the Microsporangium is called Microsporogenesis. You already studied that inside the Microsporangium, the sporogenous tissue is present, and each cell in the sporogenous tissue is a diploid cell. That cell is called pollen mother cell or PMC. And the pollen mother cell undergo meiosis, producing 4 haploid cells. 4 haploid cells in the form of an association. This is called microspore tetrad. Tetrad means association of 4 cells. Then after that, each cell will separate from each other so that separate microspores are produced. All the microspores are haploid. During the separation of microspores, they will be covered over by envelopes. They are covered over by 2 types of envelopes. Okay. The outer envelope is called XII and the inner envelope is called indine, that we study later. Anyway, this is the summary of formation of microsporogenesis. You see, microsporogenesis is the process of microspore formation. Formation of microspore within the Microsporangia anther. The diploid cells of sporogenous tissue develop first into microspore mother cells, otherwise called pollen mother cells. In short, they are called PMC. PMC undergo meiosis producing microspore tetrad. Each consists of a cluster of 4 haploid cells. These are the cluster of 4 haploid cells. As the anthers mature and dehydrate, they dissociate from each other and develop into pollen grains. Microspores are otherwise called pollen grains. Okay? Then these pollen grains produced inside the Microsporangia will be released by the dehiscence of anthers. Okay. Now we are moving on to the next prime point, Megasporogenesis. Just like Microsporogenesis, Megasporogenesis is the process of formation of megaspore. Megaspore formation takes place inside the Megasporangium. Okay. Megasporangium is ovule. The formation of megaspores within the Megasporangia of carpels is called Megasporogenesis. Ovules generally contain a single megaspore mother cell. Suppose this is the ovule, and inside the ovule, this is ovule or Megasporangium, and inside the ovule there is a peculiar type of food material, a nutritive tissue. That tissue is called what? Nucellus. And inside the nucellus there is a cell, diploid cell. The diploid cell is called megaspore mother cell. MMC means megaspore mother cell. Megaspore mother cell is diploid. When the megaspore mother cell, diploid megaspore mother cell, divides by meiosis, 4 cells are resulted. 4 cells are resulted. And 4 haploid cells are resulted in a linear manner. So they are called a linear tetrad. Out of these 4 cells, only 1 will become the megaspore and the other 3 degenerates. Okay, that's a very important point. Out of the 4 cells, only 1 will become the megaspore, functional megaspore, and 3 others will degenerate. These will degenerate. So that the process is called monosporic development. It's an important question. Monosporic development means out of the 4 daughter cells which are produced in the form of linear tetrad, only one will become functional and the other three become degenerated. So that condition is called monosporic development. Monosporic development is a characteristic feature of angiosperms with regard to megasporogenesis. So here, MMC undergo meiosis producing megaspore tetrad or linear tetrad, each consisting of a cluster of 4 haploid cells. This one. Of these, only one cell will mature into megaspore or embryo sac and the other 3 degenerate. This kind of embryo sac formation is called monosporic development. Okay. The next prime point is pollen grain. Pollen grain is the male gametophyte. What is a male gametophyte? A structure which holds the male gametes is called male gametophyte. The structure which contains the male gametes is called male gametophyte. Pollen grains are the male gametophyte. Each pollen grain has 2 envelopes, an outer envelope and inner envelope. Two envelopes are there. The outer envelope is called XII. The inner envelope is called indine. The speciality of XII is that it is made up of a very special type of chemical. That chemical is called sporopollenin. It's the most resistant biological material. And the inner wall is called indine, which is made up of pectin and cellulose, pectocellulose. The XII is porous. Some openings are there. The openings present in the XII are called germ pores, where sporopollenin is absent. But indine is non-porous. So inside this indine, you can see there is a large cell. And this large cell is a nutritive cell, and this cell is called vegetative cell. This is haploid. Then in addition to the vegetative cell, a small cell will be there. That cell is called generative cell. It is called generative cell. That also is called a haploid. Later what happens is during maturation, the generative cell divides into 2 microgametes. Okay. 2 microgametes. Microgametes are the male gametes. So when there is a vegetative cell and generative cell, that is a 2-celled condition. And when there is a vegetative cell and 2 microgametes, it is a 3-celled condition of the pollen grain. So you can see that during maturation, each pollen grain contains a vegetative cell, bigger cell with abundant food reserves, and a generative cell, smaller cell with floats in the vegetative cell. Later, the generative cell divides, producing 2 microgametes or male gametes. Thus, a fully mature pollen grain has 3 cells. Which are the 3 cells? One is vegetative cell and the two microgametes. And how many nuclei will be there? Vegetative cell has a nucleus. Each microgamete has also 1 nucleus each. So that 3 cells and 3 nuclei, all of them are haploid. And this is the structure of pollen grain. And pollen grain is considered to be the male gametophyte. Okay. Next prime point is very important for examination, that is the embryo sac or female gametophyte. Embryo sac is the megaspore. As the name implies, it is a larger cell. And the nucleus of the megaspore will divide, the haploid nucleus will divide thrice continuously. So when one nucleus divides once, 2 nuclei will be there. When it divides twice, 4 nuclei will be there. When it divides thrice, 8 nuclei will be there. Out of the 8 nuclei, 3 nuclei will be seen at the micropylar pole. 3 nuclei will be seen at the chalazal pole. And 2 nuclei will be seen at the center. The nuclei which are seen at the center are called polar nuclei. The 3 nuclei present at the micropylar pole get converted into a large egg cell. This is a egg cell, and two smaller synergids. These are called synergids. And the three haplonuclei present at the chelacal pole get converted into three independent cells, and these three independent cells are called antipodals. These are called antipodals. So this is the chelacal pole, and this is the micropylar pole. And the larger cell is called the central cell. This is the central cell. And this embryo sac is completely covered over by two envelopes, outer integument and inner integument, which has a perforation. That perforation is called micropyle. Now we can read that note. Nucleus of the megaspore divide thrice, producing 8 nucleated structure. 3 nuclei can be seen at the chelacal pole, which later form cells called antipodals. This will be. 2 nuclei remain at the center of the central cell called polar nuclei. These are the polar nuclei. 3 nuclei can be seen at the micropylar pole, which later form cells and produce egg apparatus. What is egg apparatus? The egg cell and the 2 synergids are collectively called egg apparatus. It's called egg apparatus. Egg apparatus consists of 1 egg cell centrally located and 2 synergid cells which are laterally located. Thus, a typical angiosperm embryo sac is 8 nucleate and 7 celled. That's a very important statement. 8 nucleus, but only 7 cells. Why there are only 7 cells? Which are the cells? You can see 1 central cell. Okay. 1 egg cell. Synergids 2. Antipodals 3. So that total number 7 cells. But here antipodal has 3 nuclei, synergid has 2 nuclei, egg cell has 1 nuclei, but the central cell has 2 polar nuclei. So that total number of nucleus will be 8. Okay, that is why the embryo sac is considered to be 7-celled and 8 nucleated. Clear? Now come to the last prime point of this session. With regard to pollen grain, 4 important minor points are there in the textbook. They are called pollen allergy, pollen bank, pollen viability, and pollen tablets. Minor questions may be asked from this topic. What is pollen allergy? The pollen grains of some species may cause severe allergy and bronchial problems. That means the respiratory problems leading to disorders such as asthma, bronchitis, etc. Okay. So example is very important, Parthenium or a carrot grass. The allergy caused by pollen grains of Parthenium in the respiratory tract is called pollen allergy. Next point is pollen bank. What is pollen bank? Pollen bank means the storage of pollen grains in liquid nitrogen at minus 196 degrees centigrade. This is considered to be a method of cryopreservation. Cryo means sub-zero temperature. We are maintaining, we are storing pollen grains in a highly reduced temperature of minus 196 degrees centigrade in a medium of called liquid nitrogen. So it is called pollen bank. The stored pollen grains can be called pollen banks and are used in plant breeding program. Okay. Then pollen viability. As you know, the pollen grains are released by the dehiscence of anther. The period for which the pollen grains retain their efficiency even after they are released from their anther is called pollen viability. In different plants, the pollen viability may be varying. In rice and wheat, the pollen viability will be up to 30 minutes after their dispersal. But in families Rosaceae, Leguminaceae, and Solanaceae, etc., the pollen grains retain their viability for many months. So the period for which the pollen grains are live, capable of fertilizing the female flower, is called pollen viability. Last point is called a pollen tablet. What is pollen tablet? Pollen grains used as food supplements. As the pollen grains are rich in nutrients, the vegetative cell, they can be used for food supplements in the form of tablets or syrups. So that is called a pollen tablet. And these are used to improve the performance of race horses and athletes. So 4 minor points are there: pollen allergy, pollen bag, pollen viability, and pollen tablets. By this, we are ending the first session of the chapter Sexual Reproduction in Flowering Plants. Okay, thank you.